So this is the senses chapter, and I decided to update it. The notes I have on D2L right now are kind of old, and there's lots of holes in them. So we're going to use what came with the book, and I'm going to try to refine it and make sure it makes sense. So the first part of this is about general receptors. Okay. So receptors, what they're going to do is they're going to take in information from the inside and the outside, right? So internal and external environments. The idea is there's a stimulus, and then they're going to try to decide what to do with the stimulus. So lots of times the stimulus has to be converted into a sort of electrical energy so that our cells can understand what's actually going on. So it has to be converted into a form that the cells can interpret. Okay, so then the central nervous system is going to finally decide what's going to happen with it. Okay, so receptors are going to take information to the central nervous system, and it's going to be sensory neurons that are taking that information in. Okay, so receptive fields are, the idea is that we have nerve endings distributed throughout our body. In some areas, we have a small receptive field. In other areas, we have a large receptive field. So in a small receptive field, you're going to feel more things, like if your fingers have lots of neurons and small receptive fields. So you can pick up little fine things like little beads or beans. Whereas in our back or in the back of our legs, they have very large receptive fields, so we're not as sensitive. You could be sitting on your pencil or your phone and not know it. Okay, now, a sensation is defined as a stimulus that we're actually aware of. Okay, so for us to be aware of it, it has to get to the cerebral cortex. And not all of the stimulus gets to the cortex. If we might lose it if everything got to our brain and to our cortex and we had to filter it out. Okay, so sometimes things just go to the brainstem and we don't think about it and our body responds. Okay, so there's just all sorts of different levels of interpretation of the stimuli. Okay, now modality is the type of stimulus that is coming in to the brain. Okay, so our body knows the path that the type of information should come along. So here's a really good example. Okay, so the brain, when things are coming in the optic nerve, okay, so the optic nerve is coming from your eye, that information is going to be perceived as visual because it's coming in that path. Okay, the cochlear nerve is going to go to our ear, and that's going to be like, oh, that's hearing. Okay, so things are coming in certain pathways, and that's how our brain, that's one way our brain can determine what it is, what the information is. Don't worry about this. How intense the stimulus is can be determined by how often the nerve is sending the signal. So if it's a strong signal, it's almost like the volume is turned up and it's going to be pinging more often. Okay, now these are a couple interesting concepts. Okay, so adaptation helps us kind of maybe filter out things that are around us. So we become decreasingly sensitive to continuous stimuli. So like if you live in the city, you become less sensitive to the traffic or the fire truck or the sirens, right? So you become less sensitive, you adapt. Or if you get into the elevator with someone and they have on a bunch of cologne, right? Maybe by the time you get off the elevator, you don't smell the cologne anymore. Your body adapts. So that can be a good thing or it could be a bad thing. Okay, now, two different vocabulary words. So tonic receptors, they don't adapt very fast, okay, and they're responding continuously. Okay, so in our ear, our head position, those are tonic, so they don't adapt. They're always sending a signal, so we know where our head is in space. Pain receptors don't adapt, okay? Phasic receptors adapt rapidly, so like a pressure receptor. So when you pull your ponytail tight, okay, in the beginning, when you pull your hair back and it's tight, okay, that's a new stimulus. You're going, oh, that hurts, okay, but then over time, you don't feel it anymore, even though it's still tight. Okay, now receptors are classified, sensory receptors are classified two different ways. So there's general and there's special senses. Okay, so the special senses are the five big ones. So smell, taste, sight, hearing, and equilibrium. Okay, then general senses are how you, pain, pressure, where you are in space, what you're feeling if somebody touches you. Okay, so general and special. Okay, so they're also classified by where the stimulus originates. So there are these things called exteroceptors. So that's information coming from outside. Interoceptors is information that's coming internally. And then proprioceptors, I think we talk about these last two the most. Proprioceptors, they're going to detect where your body is in space. So there's receptors in your muscles, in your tendons, in your joints. And when they go off, that helps you figure out like if your arm is up or if your arm is down or if you're like how you're standing or if you're upside down, that kind of thing. Okay, they're also categorized by how the stimulus, what kind of stimulus it is. Okay, so there's 5 types of these. Okay, so chemoreceptors are going to detect chemicals. So the chemical usually has to be dissolved in a fluid. Okay, there can be thermoreceptors, so that's going to be temperature. Photoreceptors are going to detect visual things like light, color, movement. Okay, mechanoreceptors, so that's those are things that they have like a mechanical aspect to them. So they're going to detect stretch, touch, some in your inner ear. Okay, and then the 5th one are nociceptors, and nociceptors detect pain or things that hurt. Okay, so 5 different modalities of stimulus. Okay, so chemical, temperature, light, okay, mechanical, and then pain. Okay, so tactile receptors are touch, right? So they're going to be in your skin and in the mucous membranes. Their endings can have a capsule on them, or they might not have a capsule on them. So they could be a free nerve ending, or they have this, they're wrapped up. Unencapsulated ones, those are the simplest kind of touch receptors. They're in your skin and in the mucous membranes on the surface. They're usually for pain and temperature, but maybe if someone lightly touches you. Okay, then we also have root hair plexuses. They're around the hair follicle, and so they're deeper in the skin, deeper in the dermis, and they're going to tell if the hair is moving. So like if an ant is crawling on your arm or something, it moves the hair, and then the hair sends the message that something's on there. Okay, another set of unencapsulated tactile receptors are tactile discs, and they're going to respond to light touch as well. Okay, now referred pain is in this part of the book, and we've kind of talked about it before. So this is when we talked about it a little bit with dermatomes. So this is how when we don't, our brain doesn't actually process the sensory information, right? So sometimes signals from the internal organs can be perceived as coming from your skin or your muscle, because those are the paths that are most frequently traveled. Okay, so the somatic and the visceral sensory neurons, the information is going to travel the same path. So our brain is unable to determine if the information is coming from an organ or if it's coming from the skin, because it's traveling from the same path. So why referred pain is important is because if the pain is coming from an internal organ, but our body thinks it's coming from the external part, it could mean something bad is happening. Okay, so heart attack pain, sometimes people say that their chest hurts or that their arm hurts. Okay, the kidneys or the ureter could be coming from the bottom of the abdomen. Okay, so the bladder pain could be going around and it might feel like it's in your butt. Okay, so that it can be important if someone says they have pain in a certain place and it might not be the same thing. So appendix is more towards the medial side of your body, or the lateral side of your body, I should say. But the referred pain for it is the belly button. And that's not where it is, right? Liver and gallbladder pain could be, people think they might feel it in their back or in their upper abdomen or in their shoulder. So it's not, it's kind of funky, right? Okay, so I took a lot of the clinical views out, but that's interesting information to read in the book. I left phantom pain in there. So phantom pain is the sensation that can occur when a body part has been removed. So if a limb's amputated, the sensory neurons, part of the path is still coming in from them, and the body seems to think, or the body perceives that there's pain there. Okay, so now we'll do smell. Okay, so smell is called olfaction. So it's going to detect odorants. So odorants are just molecules that are in the air. Okay, so smell is actually really important. It helps keep you alive because of different odors. So odors could be dangerous. Okay, could keep you from eating bad food. So the olfactory epithelium is where we detect the odors. And it's going to be up here. So here's a mid-sagittal cut through the head. Okay, and odorant molecules are going to come in. Okay, and then up here is the epithelium. Okay, and you don't have to know all of these parts, but what I want you to understand is there are holes, little tiny holes in the skull. Here is the olfactory bulb, so that's cranial nerve number 1. Okay, and nerves extend from it down into this mucous membrane. Okay, so here's the skull, right? Here's the optic nerve. Okay, and then this extends down through here, and then you have these nerve endings in your nasal passage. Okay, those nerve endings, okay, detect the odor molecules, and then the information is going to be sent up into the brain. Okay, these actually are replaced every 40 to 60 days. So they're in there. If you think about your nose, right, your nose is exposed. So those cells will go through mitosis and will be replaced. Okay, so I know I'm not too worried about this. I mean, you should know olfactory nerve is cranial nerve number 1. Okay, the olfactory bulbs, they're paired, right? And I'm not worried about that. I'm actually not worried about that. Okay, don't worry about that. I should have taken that out, but I didn't. Okay, so the mucus in your nose has proteins that are going to bind the odorants. Okay, and then the information is going to go to the central nervous system. Okay, so it could go to the cerebral cortex where you're going to figure out what the smell is. The hypothalamus might cause some sort of gut reaction to the smell. The amygdala is going to help you figure out what the smell is and maybe trigger some sort of emotional response. Okay, so you could think about chocolate chip cookies, right? And maybe smelling them and having a good response, or something, or the dead cat in anatomy lab, right? And that could be a visceral response. Okay, now taste, gustation is taste. Okay, and the taste molecules they call tastants. The cells there's chemoreceptors in the taste buds, so they're in the papilla of the tongue. There's a couple different kinds of papilla on your tongue. There's some that are short and spiked, and they're called filiform. And there are some that are called fungiform. Okay, there are also foliate and valate. Okay, the valate have most of the taste buds. I am not too worried that you memorize the different kinds of taste buds or the different kinds of papilla on the tongue. So don't worry about these. There's a lot of bigger stuff to worry about. Don't worry about that. So this picture tries to show you what the different taste buds look like and where they would be found on your tongue. Okay, there are 5 basic tastes, and you guys can read them. They're sweet, salt, sour, bitter, and umami. Okay, and I'm not too, there's more important stuff to worry about than taste, so don't worry about this too much, right? Just know it exists. There's 5 different taste sensations. It's happening with there are papilla on the tongue. The one that has the most taste buds, they're called the valate. Okay. Don't worry about that. Okay, now the eye. Okay, so the eye we're going to start on the outside with the accessory structures. Okay, so the accessory structures that are around or attached to the eye, so are the eye muscles, the eyebrows, the eyelids, eyelashes, conjunctiva, okay, and the lacrimal glands. So we're going to talk about kind of all of that. Okay, so if we look at this picture, which to me the font is small on this. Okay, so here is an eye. If we did a sagittal plane and we cut through the eye, okay, so here's eyelashes, right? And they're going to protect the eye from things going into there. So is the eyebrow. Let's just kind of go through the parts. Okay, so here's this outer layer, that's the cornea. Okay, no, you don't have to know that. We'll go over this later, the orbicularis oris. We'll go over it. We don't worry about that. Don't worry about that. The tarsal plate is where it's a sebaceous, so there's going to be oils coming down. So if you get it, and there's tarsal glands, so if someone gets a sty in their eye, the tarsal glands are infected or blocked. Orbital fat is going to keep your eyeball in place. Okay, and we'll talk about this muscle more later. Okay, the pupil is the hole in the eye that the light is going to go through. The lens is malleable, so it moves. Okay, so the lens is what's going to focus the image as it's coming in. The iris is the colored part of your eye. Cornea, right, is clear, but the sclera is the white part. Okay, so the sclera is the hard white part around the eye. Okay, so the lacrimal apparatus is tears. Okay, so it's going to produce, collect, and drain fluid. So in the tears, you have water, you have salt, you have antibodies, and you have lysozyme. Lysozyme is antibacterial. It's a lysing enzyme. It's going to help protect your eyes from bacteria, okay, or viruses. The tears, their purpose is to lubricate, clean, keep your eye moistened, and reduce friction. It's going to help protect you, and it's going to give oxygen and nourishment to the cornea. Okay, the lacrimal gland is what produces the tears, and we blink between 15 to 20 times a minute. Okay, so the fluid from the lacrimal glands drains into little holes called lacrimal puncta, and then those are going to drain and then go into the lacrimal sac. Okay, the sac then drains into the nasolacrimal duct to the nasal cavity. Okay, so here's the gland. Okay, and then the tears are going to get released. Okay, and I'm not, the puncta is the little holes, right? There's the duct. Okay, and that goes down into the lacrimal sac. Okay, so the tears will accumulate up here. They're normally just released through here, and then they're going to drain down into your nasal cavity. Okay, so the eye is like 2.5 centimeters in diameter. Okay, there's fat around it to keep it in the skull in place. Okay, there's 2 cavities in it. The cavity that's behind the lens is called the posterior cavity, and it contains a fluid that's called the vitreous humor. Vitreous humor is permanent. We're born with it. We keep it. The anterior cavity is in front of the lens, and it has aqueous humor. Aqueous humor is renewed. It circulates. There's 3 layers in the wall of the eye. So there's a fibrous tunic, which is the outer layer. So if we go like this, there's fibrous, there's vascular, which I'll make red because vascular is blood, right? Which is the middle. And then the retina, let's just make it, I don't know, light blue. And then the retina is the nervous part, and that's on the inside. Okay. So the sclera is fibrous, the vascular layer is the choroid, and the retina is the innermost layer. So they say scared cats run, to remember those layers from the outside in. Okay, so here it is again. So the fibrous layer, the outer layer, is the sclera and the cornea. The vascular tunic is made up of the choroid, the ciliary body, okay, and the iris. And then the retina is this is the neurons. This is the part that does the work. It's going to have you see. So sclera is the white. Okay, and the cornea is the front of your eye. Okay, now if we look inside the eye, hopefully you guys can see this. Okay, so here's the lens, right? So behind the lens is the posterior cavity. So that's where the vitreous humor is. To the front of the lens is the anterior cavity, and that contains aqueous humor. Here's the optic nerve coming out the back. Okay, and where the optic nerve leaves is called the optic disc. And the optic disc is actually where we have the blind spot in your eye. Okay, so that's where it's leaving, so there's no photoreceptor cells there to actually see. Okay, now there's a place on the back of your eye, it's called the fovea centralis. And in our eye, we have rods and cones. Okay, so rods and cones are the photoreceptor cells. So those are the parts that do the work, that allow you to see. So the fovea centralis this is where we have the clearest vision. Okay, and it is a place where we have rods and cones. Okay, every place else we have just rods. Okay, so rods are for black and white vision, and cones are for color vision. So C and C. Okay, now there is a wrinkly edge here where the retina comes together, and that's called the ora serrata. Okay, there's a ciliary body. So the ciliary body and these ciliary processes are attached to the suspensory ligaments, and that's what's going to hold the lens in place. So the lens is going to change shape depending on how you need to focus. Okay, and then I think I went over the other parts, right? We did the pupil, we did the cornea, the iris is the colored part, the lens. I'm not worried about the limbus. The scleral venous sinus is an opening, and that's where the aqueous humor is going to drain. I'm not worried about the sphincter pupillae or the dilator pupillae. Okay, but that's eye anatomy. Okay, so the sclera is the white of the eye. It's where the external eye muscles attach. It's going to keep the eye shape. Okay, the cornea is the transparent window, right? It doesn't have any blood vessels, and I think that's good. Okay, the vascular tunic is the choroid. Okay, so the choroid, well, there's several parts of it, right? So there's the choroid, there's the ciliary body, and there's the iris. So the choroid lays on top of the sclera. Okay, so let's say this is the sclera. The choroid lays on top of the sclera, and it nourishes the retina. Okay, so the retina cells are in here. Okay, and they don't have their own blood supply. So they're going to get nourishment and waste taken away from the choroid. Okay, it's dark looking. Okay, so it has melanocytes, and the dark is going to absorb the extra light. Okay, now the ciliary body has muscles in it, and it's going to help change the shape of the lens. And it's also going to be where we make aqueous humor. Okay, another part of the vascular tunic is the iris. And the iris is going to control the size of the pupil. Okay, and the pupil's a hole. Okay, so the pupillary reflex. Okay, so if we're in low light, the pupil's going to dilate and get bigger so more light can get in and you can see. Okay, so that's sympathetic nervous system. And parasympathetic, when we're relaxing, it's going to constrict the pupil so we don't need as much light. Okay, so now the retina, there's several layers of the retina. Okay, so there's the pigmented retina, and it lays right on top of the choroid. Okay, the neural layer is where we have the photoreceptors. So that's where we have the rods and cones. And then the ora serrata, it's just this jagged edge around the eye between the photosensitive and the non-photosensitive parts. Yeah, it's a serrated edge. Okay, now the retina is made up of several different types of cells. Okay, so the photoreceptor cells are rods and cones. So they're going to react to light. The bipolar cells are going to take information from the rods and the cones. Then the ganglion cells are going to come together and they're going to form the optic nerve. Okay, I am not worried about these other retinal interneurons. Okay, so here's kind of a picture trying to show you incoming light. Okay, so the shallow line is supposed to be light coming in, and this is how the information is going to get out. Okay, so here's the photoreceptor cells, okay, the rods and the cones. Okay, so you see these cones look like they have little cones, and the rods look like this, okay? Then the information is going to come to the bipolar cells, and then it's going to go to the ganglion cell, and the ganglion cells can take it to the brain. But the light's traveling in that way. All right, also in the eye, and I mentioned this when I was doing the anatomy, contains the optic disc, okay? So that's the blind spot. The macula lutea is close to the optic, it's beside the optic disc. So if this is the back of your eye, okay, here's the optic disc. And then over here, kind of, this is the macula lutea. Okay, and it has a thing in the middle of it that is called the fovea centralis. Okay, the lens. Okay, so the lens doesn't have any organelles. Okay, it's kind of dense and it can change shape. Okay, so when you're looking at something that's more than 20 feet away, the lens is flattened. When you're trying to look at something up close, okay, the lens accommodates and it becomes more spherical. So it's trying to focus. Okay, so this is what this is trying to show you. So it's flat and it's more round. Okay, the fluid that's in the eye. So in the posterior cavity, we have vitreous humor, and it's kind of thick and gloppy, almost like maybe gelatin. We have it all the time. It's going to keep the retina flush against the back of your eye. Okay, so it's going to keep the retina up against the choroid because the retina it's not really anchored on there. It could come off and flop around. Okay, the aqueous humor is to the front. It's in the anterior cavity. Okay, it's going to feed the lens and the inner part of the cornea. Aqueous humor, here's a picture. Okay, so aqueous humor is going to be produced in the ciliary processes, okay, from the capillaries. And then it's going to go through the pupil into the anterior chamber of the anterior cavity, and then it drains through this scleral venous sinus. And then it's going to dump into the bloodstream at the veins. If it doesn't drain right, the pressure can build up in the eye, and that can lead to glaucoma. Okay, so this picture is trying to show you how it flows. So here's one. Aqueous humor is secreted by the ciliary processes into the posterior chamber. Okay, so here it is. It's being dumped into here. Let me use the pointer. Dumped into here. And then it moves from the posterior chamber to the anterior chamber. Okay, and then it can get picked up at these scleral venous sinuses so that it dumps back into the blood. If these get clogged up, then the pressure can build up and it can put pressure on the retina at the back of the eye. I think I kind of, let's, how our eyes focus. Okay, so if we're looking at things closer than 20 feet, the eyes have to converge and the lens is going to change shape and the pupil is going to constrict so that we can see it. The extrinsic eye muscles have to do that to pull things in, and the ciliary muscles has to contract and thicken the lens, and the pupil has to get smaller. If objects are further away, the eyes are not going to be converged. The lens is flat and the pupil is dilated. Okay, now the process of taking the light and turning it into electrical signals is called phototransduction. Okay, rods and cones are what does that. I don't usually make anybody memorize these photoreceptor parts. I'm not worried about that. Okay, so rods are longer and more narrow than cones, and there's more of them. Okay, they're really sensitive, even if it's dark. Okay, cones are concentrated at this fovea centralis, and cones have to do with color vision. Okay, cones have a one-to-one relationship with the bipolar cells. Okay, so they're going like this. Many rods, okay, go to one bipolar cell. Okay, so this is more, if you were just left with rods, your vision wouldn't be as clear. Okay, but if you were only left with cones, you would have a sharper vision. Okay, and that's just a picture of that. Okay, now, phototransduction. How does it actually work? Normally, I don't make you learn this because there's so much other stuff in this chapter. So don't worry about that. Okay, now, the visual pathways. Okay, so the information is going to come from the rods and the cones to the bipolar cells to the ganglion cells. And then the ganglion cells are going to come together and they're going to form the optic nerve. Okay, then the optic nerves are going to cross, right? And that's going to go to the thalamus. And then from there, it's going to go to the occipital lobe of the brain. So that's kind of the pathway. Our eyes have overlapping visual fields. Okay, so it allows us to have depth perception. The superior colliculi of your brain are going to coordinate your eye movement. The pretectal nuclei are going to coordinate how your pupils respond and if your lens accommodates. Okay, and then I think, all right, I'll go ahead and do ear. I'm just trying to decide whether to make this all one big lecture, but you guys can stop it if you need to. Okay, so ear. So the external ear is called the auricle. Okay, so it's the outside of the ear. And then the ear canal is called the external acoustic or auditory meatus. Okay, the cerumen is what is the technical name for earwax. Earwax is there because it keeps microorganisms from growing. Okay, the tympanic membrane is the eardrum. Okay, so if we look at this picture, we can kind of hit everything on it, I think. Okay, so there's the auricle. Here's the bone part of it, right? And then within the bone, there's all sorts of membrane. One interesting thing or important thing to know, okay, here's the external ear, and the external ear goes from the auricle into the tympanic membrane. Okay, the middle ear is going to be from the tympanic membrane back. Okay, and then the inner ear is the cochlea and the stuff that's within that. Okay, so sound's going to come in the external auditory meatus and it'll vibrate the tympanic membrane. The tympanic membrane will then move the auditory ossicles. Okay, so the ossicles are the smallest bones in your body, and there's 3 of them. So they go in order, they go MIS, so malleus, incus, and stapes. They are going to move and vibrate this thing called the oval window. Okay, and you can't really see what's going on, but the stapes is hooked to the oval window, and that's going to cause fluid within the cochlea to move. Okay, the entranceway to the cochlea is called the vestibule. Okay, semicircular canals are part of this organ, and they're going to help us with balance and equilibrium. The auditory tube is the inner ear tube. Okay, and this is going to go down to your nose. The vestibulocochlear nerve is cranial nerve number 8. Okay, and it's got part that's coming from the vestibule and part coming from the cochlea. So the cochlea has to do with hearing, and the vestibule has to do with equilibrium and balance. Okay, so I might have just gone through all this. So the middle ear has the tympanic cavity. Okay, it has the oval window and the round window, which will come and be important in a minute. Okay, the auditory tube, they used to call it the eustachian tube. So it goes from the middle ear to the upper part of your throat, so the nasopharynx. So middle ear infections are when you, like an earache, is when you get organisms or mucus from the nose, and it lays in this tube. So that tube, when you're little, it's flat. Okay, and then as you get older, it becomes more up and down, okay, more vertical and less horizontal. Okay, so the bones of the inner ear are the malleus. It looks like a mallet or hammer. The incus, which looks like an anvil, and the stapes, which looks like a stirrup. Okay. The ossicles, they amplify the sound waves, and it goes along to the oval window. Okay, the oval window, it's gonna, here, let me try to. So here's a blow up picture. So here's the eardrum, which is the tympanic membrane. Right, here's the inner ear, the eustachian tube. Okay, here's the malleus. Okay, here's the incus, and then here's the stapes. So 3 bones all hooked together. The stapedius is a muscle that anchors the stapes, and the tensor tympani is here too. It's cut though. All right, so now in your inner ear, okay, so it's in your temporal bone in your brain. Okay, so in there's what they call a bony labyrinth. Okay, so it's made out of bone, and it's kind of like a maze or a labyrinth, right, in the bone. And in there is fluid, and the fluid is called perilymph. Okay, now the membranous labyrinth there's actually membranes within the bony labyrinth, and there's fluid in there as well, but that fluid is different, of course, right? And that's called endolymph. Okay, now in your inner ear, you have 3 things that are going to be really important. So one is the cochlea, okay, and that's where hearing's going to occur. One is the vestibule, and that's got to do with equilibrium and the semicircular canals. Okay, so if we look at the inner ear, okay, so here's the cochlea, okay, and it looks like a snail. That's the cochlea. Here's the semicircular canals. Okay, and then this part here, which they have it kind of like there, is the vestibule. So the vestibule is the entryway. You don't have to know like all of this. Endolymph and perilymph are important because that's fluid that's in there. So there's liquid in there. It's not dry. Okay, so when the liquid moves, different things are going to happen. Okay, now 2 other things. So there's a utricle. Where's the saccule? And then there are these things called saccules. We're going to talk about those. Okay, they have to do with movement and keeping your balance. Okay, cochlea. Don't worry about that. I'm going to show you on a picture, so I'll just leave these here and I'll try to show you on this picture. Okay, here's hearing. Here's how hearing works, which I'll show you here. Okay, and then here's a cochlea. If you took a slice through it. Okay, if you took a slice through it, what you're going to see is you're going to see, this is kind of hard. There's, let me see if I have a better, this might be better to see it. Okay, so if you looked at the cochlea, so this right here is this made big. Okay, so in here, you're going to see these membranes. Okay, so here's the basilar membrane. Okay, and here's what's called the tectorial membrane. In here is where the little hairs that are going to help you hear are. Okay, down here is the scala tympani, and up here is the scala vestibuli. I know, this is hard to explain to you without seeing you in lab. Okay, so here's another picture. So here is this part that I made purple over here. Okay, so here's the bottom membrane. Okay, and here's the top. Okay, see the cells, right? And then see, these are hair cells, right? So stereocilia are these little hairs up here. Okay, so what's gonna happen is when you hear something, these are gonna move. And when they move, they're gonna transduce a signal, and the signal's gonna go out here. I'll try to find a video so you guys can watch it. Okay, so here's basically what happens. So the sound waves vibrate the tympanic membrane. Okay, that moves the ossicles, and the ossicles are gonna vibrate the oval window. Okay, so what happens then is the fluid in here is gonna move. And when that fluid moves, okay, the hairs move. So when the hairs move, okay, the hair cells are distorted, and that's going to release neurotransmitters or causes changes in neurotransmitter release, okay? Then the sensory information is going to go down cranial nerve number 8, okay? And what will happen is the leftover pressure is going to go to the scala tympani, and it gets kind of dampened so it doesn't keep vibrating. I need a video. There's probably one in the Learn Smart, and I'm sure I assigned it, but I can't remember. Okay, so this kind of shows you how sound is going to flow. Okay, so it's going to come in this external auditory meatus, vibrates the tympanic membrane. That moves the malleus, the incus, and the stapes. Okay, that's going to vibrate the fluid that's in the scala vestibuli. Okay, when that vibrates, okay, it's going to move the fluid in here, and that's going to move the hairs. Sorry. Okay, and then the neural impulse is going to go down the nerve, and it's going to go to your brain. The extra movement is going to be dampened here at the round window. So it gets absorbed, so you don't have like reverb going on in your ear. Okay, you guys can read this, I think. I just tried to explain it. Here it is all written out in words. Okay, here's the hair cells. I'm not worried that you know this. Okay, just the idea that they're little hairs. You guys can read about pitch. Don't worry about this. You can read about loudness, and then equilibrium is monitored by the vestibular apparatus. Okay, so it's the semicircular ducts and these utricles and saccules. Okay, so they're going to help us figure out if we're moving and where we are in space. Okay, so I am going to stop this here, and then I'll make a video just on this part.